I'm Gracie Lay. These are the lab work photos that I've took over the course of the STRIDE program. I'll be discussing the methods and techniques I've learned and how they apply to my research. Of the techniques and methods that I've learned, I'll be going over cell culture, immunocytochemistry, fluorescence microscopy, fluorescence analysis, and mouse brain dissection. One of the main things that I did during my time here was cell culture, specifically maintaining and passaging them. I switched astrocyte cells containing GFAP from one container to another, from a T25 to a T75. I split the cells into two T75s and put it into suspension. Overall, I was able to prepare medium changes and splitting procedures. These cultures were then used for immunocytochemistry, my next step in conducting my research project. I was also able to perform immunostaining, specifically immunocytochemistry. I performed immunostaining in order to assess the presence of GFAP in cells by use of the antibody PBS. In this image, I was performing immunostaining on glass cover slips containing the fixed cells after cell culture. Essentially, from this process, I was able to stain GFAP, which led me to the next step, fluorescence microscopy. After staining the GFAP on the cover slips, I was then able to get a closer look through a fluorescent microscope. The fluorescent microscope provided great use due to its great sensitivity for detection and quantification of small particles, yielding extremely detailed and reliable images. Specifically, I examined the intensity and expression of light being expressed by the cells on the cover slip. The microscope images allowed for the next step, fluorescence analysis, to take place. With the microscopy images produced, I was able to determine the level of GFAP expression using the software called Fiji/ImageJ. I compared the fluorescence intensities of knockout and wild-type samples. Here, I created a spreadsheet with the mean and average fluorescence activities. These fluorescence intensities indicated expression of GFAP. Essentially, the brighter the cell, the more of the protein expressed. With the analysis, I was a step further into my research. Last but not least, one of my most memorable experiences while working in the laboratory was dissecting a mouse brain. The mouse was euthanized through carbon dioxide, then decapitated for its brain. Dissecting a mouse brain was quite difficult, as it was not only small, but slimy and squishy. Though it was not an easy process, I found it very fun, as it was a new experience for me, and definitely different from what I've done at school so far. And that concludes the main methods and techniques that I've learned at the laboratory this summer. Thank you for listening.